Another type of movement is going to be vesicle movement. Vesicle. Oh. Vesicles were a structure that we talked about when we talked about cells, right? They were just little storage sacs. Vesicle movement is for molecules that are too big, all right? Too big for diffusion, too big for a protein channel, right? They're so big that they need their own mode of transportation. They need a specific method just to get themselves in or out of the cell. They need a vesicle to help. So that vesicle is a membrane-bound sac, and it's going to bring substances in or out of the cell. And a vesicle is made of a phospholipid bilayer. It's just like the cell membrane, right? So here's a picture of our vesicle. And let's say we're transporting a protein. Here's our protein packaged inside of our vesicle. Here are our phospholipids. All right, all of our phospholipid bilayer. Right? Just like the cell membrane, just doesn't have all the stuff inside like a cell would. So let's talk about movement in. Into a cell is called endocytosis. We can take this word and we can break it down. Endo, meaning in. Cyto, meaning cell. Osis, all right, or cis, meaning a process of. So this is a cellular process moving stuff in. All right, this is how cells get moved in with a vesicle. I also tell my students endo for enter. All right, this is the way in. Endocytosis is for entering. You got two types of endocytosis. Penocytosis. Right? Penocytosis is when liquids, all right, are going to be packaged and moved in or out of the cell, all right? This is like cell drinking. And then you've got phagocytosis. Phagocytosis is for food. This is how food molecules, large solid molecules, are going to be moved. So we've got penocytosis for liquids moving in, phagocytosis for solids moving in, all right? So we equate them to cell drinking and cell eating. This picture right here is showing you endocytosis. You have all of your little liquid molecules. These liquid molecules are not water. Why? Water moves by osmosis, right? Not endocytosis. All right, so here are all of our little liquid molecules. They're going to push up against the cell membrane. Cell membrane ain't letting them pass through. They're too big to diffuse straight through the cell membrane. So the cell membrane starts to actually dimple in. That dimple starts to form a pocket. That pocket starts to actually close in on itself until it closes completely and pinches off a vesicle. Now that vesicle is inside the cell, endo, entering, it's inside the cell, and the stuff inside of it has gotten in. Now we said that the membrane of a vesicle is still a phospholipid bilayer. Well, look at it. Where did it come from? It's a part of the cell membrane that broke off, right? So it should be made of the exact same stuff as the cell membrane. Phagocytosis, showing the same thing, except for now we're looking at a solid. Right? This big giant molecule pushes in on the cell membrane, creates a dimple, a pocket, and that pocket pinches off to create a vesicle inside. Let's say that's a big protein, right? You had eggs or chicken or something that you ate, and you've got a lot of protein. That protein has come into your cell, right? Now it's inside of your cell, and it needs to be further broken down, right? Because your cells can't use whole protein. Your body doesn't require the same proteins that a chicken's body would or that a cow's body would, right? You require different stuff. So you need to first disassemble your proteins, break them down further. What would be the first organelle that you would go to? First organelle that this vesicle would go to would be the lysosomes. Lysosomes contain digestive enzymes, right? The lysosome is going to break down, right? Break it all apart into all these little amino acids. Amino acids, remember, they're the subunit of the protein, right? Now you have molecules that your cell can use, all right? Amino acids are usable by the cell. Whole proteins, all right, you need to first break them further down before your body can start to use them. So now let's talk about how stuff gets back out. Exocytosis. All right, exocytosis, I like to tell my students, exo for exit. This is the way out. This is basically going to be endocytosis, but in reverse. All right, it's going to look very similar. Think about it this way. Now you have all those amino acids, right? You ate something for lunch, you ate protein, whatever, and the protein has gotten into your cells. Your cells have broken them down, and now you have all these little amino acids floating around. Those amino acids need to be assembled into something that your cell can use, right? It needs to be assembled into a protein that you actually need. Let's say the protein that your body needs is hemoglobin. Hemoglobin is the protein on red blood cells that's responsible for carrying oxygen. So, the first thing that's going to happen is that those amino acids are going to get reassembled and turned into a protein by an organelle. What organelle makes proteins? The ribosomes. So the ribosome builds a new protein, builds a hemoglobin molecule, this new protein. The ribosomes send it to the Golgi apparatus because the hemoglobin is not needed inside the cell. It's needed outside of the cell where it can carry oxygen. So the Golgi apparatus is going to package it up into a little vesicle and send it to the cell membrane, where that vesicle can leave the cell by exocytosis. So, all right, here's our picture. This would be like our protein, all right? It's not really a good color, sorry guys. This would be like our protein inside of our vesicle that's been packaged, and it's going to get a chance to leave so that it can do its job outside of the cell. Again, same thing. Phospholipid bilayer, when that vesicle comes in and merges with the cell membrane, it's just going to become a new part of the cell membrane.